This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. From Microbe TV, this is TWiM. This Week in Microbiology, episode 131, recorded on July 21st, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,400 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How are you?
I'm well today.
Last time I spoke to you was in Boston, Massachusetts.
Yeah, I barely recovered from that meeting. I think my Fitbit was recording. I was doing approximately 15,000 steps throughout the meeting, running from this meeting, the posters. And so I don't get that much exercise every day. So it was really a workout.
Good meeting.
It was fantastic. The problem I had is the dilemma of which session to go to. There was just so much good stuff that I was interested in.
Yep.
And I just had to pick and plant myself and not run around like a crazy person.
Unfortunately, I had to leave on Sunday and head down to Virginia to ASV.
Oh, the virology meeting. How was it?
It was very good, but I was only there Monday and Tuesday. I had to leave Tuesday to get to my daughter's high school graduation.
Well, that's nice. Congratulations.
And so my kids are now all out of high school. They're all going to be in college this fall. 3 kids in college. They're going to be poor.
We're going to be sending you coupons.
That's okay. It's okay. Now I can do a little more traveling during the year, you know.
Yeah.
So then after that, I went off to Japan for 3 weeks.
Ooh.
With the two kids who were just going to college, my wife, and that was a blast. I just loved it. Japan is great.
Did you take the trains?
Took the trains, and they are so good. They make the trains here look terrible. I took we took all kinds of trains, but we took the Shinkansen between big cities. They are so smooth. It's amazing. And, you know, I take often the Acela between New York and Washington when I go to ASM. And that's supposed to be our high-speed train. It's not very fast by comparison. And there are some parts of the tracks where I can't even type. It's so bumpy.
Oh, yeah.
And the one in Japan was it was like an airplane ride. It was just beautiful. Oh, well.
Yeah, I last time I was in Japan was 2007 when I was invited to come over and speak on sustainable practices in universities. And I went to Matsumoto University. Turns out the president of that university was a postdoctoral fellow here at the medical university. And I, for many years, had been running the sustainability effort here on our campus. And so he invited me to come over to speak to his faculty. So he sent a faculty member down to meet me at Narita Airport, and we took the train back to Matsumoto. And the faculty member pointed out to me to pay attention as I was riding these 5 different trains from the bullet train down to the locals, all the way out to this spectacular university. And so I paid close attention as we were changing trains in Tokyo and changing trains in this other city. And, you know, even me, the one person who has trouble deciphering Japanese characters, was able to navigate the system pretty well, because I got back and then I went to a university in Tokyo and gave a talk before I came back home.
I loved it. It's a great country. They have great technology. They have great food. They're really nice. Everything works. Mm-hmm. I had a great time. I just loved it. And It's the longest vacation I've taken in years, but it was really worth it. I figured I'm going 12, 14 hours. I might as well stay for a while.
That's right.
So I'm back, and here we are podcasting. So it's you and me, Michael. We can go on for days if you want.
Oh, my. We're going to get letters in for sure. We'll be in trouble. It'd be like the old days.
Michelle is in Europe, right?
She's in France, from what I understand.
And Elio is just traveling. I think he has a wedding or something, right?
He takes his July constitutional vacation, I believe.
So we can do this. And we have some interesting stories today, starting with two classic spotlights. Tell us about that, Michael.
Well, beginning with the January 2016 edition of the Journal of Bacteriology, it's its 100th anniversary this year. The editorial body of that journal elected to publish in each future issue a discussion of one or two influential papers that were published by this particular journal in the preceding 100 years. And as Elio would say, these are classic papers that are must-reads of everyone. And they're written in a style, and it's they not only reference the original paper, but one of the journal's editors actually writes the spotlight. And it's technically called an editorial because it's the opinion of that particular author highlighting what they do. And Tom Silhavy, who's the editor-in-chief of the Journal of Bacteriology, wrote on the anniversary editorial that he was privileged to write. He wrote, on this occasion of the journal's 100th anniversary, and now I'm quoting, these brief articles will explain why these papers were so important in ways we think will be immediately apparent even to non-experts. I think it is true today that the younger scientists do not read journals. What they rely on is electronic searches that notify subscribers of papers published in the areas of interest by email. I think young people will learn much from these classic spotlights, and I urge you to call them to their attention. Looking at the Journal of Bacteriology table of contents will be rewarding, he promises. Well, I agree wholeheartedly with Dr. Silhavy with the sentiment. And Tom is no stranger in advocating the value of literature. And those of you who were schooled in the era of post-classical bacterial genetics likely recall his Cold Spring Harbor book entitled The Power of Bacterial Genetics that he co-compiled with John Beckwith from Harvard. In this particular book that was published by Cold Spring Harbor back in 1991 was a compendium of the then greatest papers in bacterial genetics, and it was dedicated to the memory of Philip James Basford Jr., who died unexpectedly in December of 1991, the year that book was published, and John Schaaf, who died earlier that year in May. And both of those individuals had collaborated with both Silhavy and Beckwith, and they were both outstanding bacterial geneticists. So I was scanning the Journal of Bacteriology contents, and I stumbled across this, and I knew Phil Basford, and I know Dr. Silhavy, and I know John Beckwith, and so I said, okay, I'm going to see what this classic spotlights are all about. So the first paper that I selected for our snippet today is one of these spotlights entitled To the Periplasm and Beyond, Protein Secretion in Escherichia coli. And this paper highlights a work by Basford, Silhavy, and Beckwith, and was entitled Use of Gene Fusion to Study Secretion of Multibiodine Protein in E. coli's Periplasm. And it was published in July of 1979. In an era when JBAC only had monthly releases. And so the spotlight, or this perspective/editorial, was authored by Conrad Molyneux from the School of Biological and Chemical Sciences at Queen Mary University of London in the UK. So here is the snippet. This paper was one of a series of studies in which gene fusions. Now, the gene fusions that we're all familiar with typically involve, jellyfish proteins. But this was good old beta-galactosidase. And anyone who's ever used a blue gene selection or used XGAL, but this was in the era before XGAL, and these were using ONPG, which is orthonitrophenylthiogalactoside, which would turn yellow when you would do an enzyme assay. And it was the old Miller assay. The other Jeff Miller pioneered that assay. And that's in one of the famous Cold Spring Harbor methods books from Bacterial Genetics. And so this particular reporter construct was the gene for beta-galactosidase, which is an enzyme that cleaves lactose to the monosaccharides glucose and galactose. And Basford made elegant use of this enzyme, which many of you know is active only in the cytoplasm of bacteria. And the other thing you have to keep in the back of your mind in order to appreciate the elegance of this selection is that in order for beta-gal to work, it requires tetramerization. That is, four of these proteins must come together in order for it to have its activity. And so what he did, or what Silhavi, Basford, and Beckwith did, is they fused the LAC-Z gene to the maltose binding protein gene, or the MAL-E gene. And maltose binding protein, or I'm just going to refer to it hereafter as MBP, is actually targeted to the periplasm of the bacterial cell. And this is a protein that has a gift. It has what we now know as a signal sequence. But the mature protein is 38,500 Daltons. And when the signal sequence is cleaved off of this microbe, it's, of course, shorter. So you can tell what's inside a cell and what's outside of a cell by using an antibody to MBP, precipitating it with Staph A, and you can then run it out on a polyacrylamide gel. And if you radiolabel your proteins like they did back in the ancient times with S35 methionine, you can actually see where the protein is just by looking at it on a gel. The ones that are in the cytoplasm are bigger because they still have the signal sequence attached, and those that are properly exported to the periplasm are shorter, or about 38K in length. Now, MBP also serves as the maltose chemoreceptor. So you can actually turn on the whole maltose catabolism system, because it's an inducible system in bacteria, through the addition of maltose to the medium. And MBP is actually the receptor that accepts the maltose and says, okay, turn this on. Now, many of you already know the classic signal sequence and how it works, but what the cell actually does is it appreciates the destination of maltose binding protein through the interpretation of the octanol water coefficient of the first few amino acids as they begin to emerge from the ribosome. And again, in bacteria, transcription and translation are coupled. And as the ribosome is beginning to push that protein out, the signal sequence will actually be targeted. And subsequent papers that were just but a glimmer in Beckwith's mind back in the late 70s and early 80s effectively have given us the canonical general protein export mechanism. But back to the data in this particular paper and why I think Molyneux selected. The signal sequence at that time was thought to initiate passage of the polypeptide through the membrane while it's still attached to the ribosome. And then the protein leaves the cell via the general export system of bacteria, or the sec translocase, which we now know because that is, again, another subsequent paper by Beckwith's family, and that happens to be a paper by Oliver and Beckwith that came out in 1981 that Molyneux also brings to our attention as we read through this nugget of wonder.
Now, so Oliver is the guy you worked with, right?
Yeah, Don Oliver is the one I work with. So I'm actually the grandson postdoc that began to work on these things. So I was actually, when I joined the lab in 85, I had read all of these classic papers that were not yet classic. They were the basis of the work that I was starting. So
Oliver discovered Sec-A, right?
Yes.
In 1982, Oliver, and that must have been he must have shortly after that gone to Stony Brook, right?
Yes, that was his postdoc project.
Yeah.
And so what's really neat about this is the gene fusion technology that Basford and actually Beckwith then, pioneered, and a lot of people have used these gene fusions. And there's, of course, gene fusions where you fuse the protein together, and that's really the key to this selection, is that beta-gal or the LAC-Z gene was fused in frame to maltose binding protein, so you make a hybrid protein. And because that hybrid protein gets stuck, it doesn't actually completely export. And Mulino points out we still don't know why X-gal can't be exported from the bacterial cell. And my suspicion always has been it's because it tetramerizes readily because its activity is cytoplasmic. And so you have four things trying to go out. You have four people growing out a door that's only intended for one. And so this hybrid protein only includes a small terminal portion. And again, as you go back and you read Beckwith's original paper, today graduate students would make these constructs very differently. They would order the sequence from the oligo person, and they would literally make the genes in frame and PCR them up, and you'd have it all done over the weekend, and you'd come back and you'd show your advisor what you have accomplished over that weekend. But what they had to do is they had to do classic genetic screenings, and it's just a joy to reread this paper. And so I'll just take you through the basics of the paper. When the hybrid protein only includes a small amino terminal portion of MVP, the hybrid protein resides in the cytoplasm. And so you have then X-gal or beta-gal activity. When the hybrid protein contains enough of the maltose binding protein to include an intact signal sequence, a significant portion of that hybrid protein is found in the cytoplasmic membrane, suggesting that secretion of the hybrid protein has been initiated. And again, the trick they use is they can see where it is based on the presence or absence of the signal sequence on that polyacrylamide gel. In no case is the hybrid protein ever completely secreted into the periplasm. And here's where luck actually paid off, is using the maltose binding protein in concert with X-cal. And so in the latter case, the synthesis and attempted export of the hybrid protein interferes with the export of at least certain envelope proteins, which then accumulate in the cell in their precursor forms. And because secretion is an absolute behavior, simply because bacteria put so many of their proteins in the membrane, the periplasmic space, and the outer membrane in order to get the business of the cell done, if all of these things are stuck, the cell effectively, it's like a traffic jam, or everybody trying to go through the Holland Tunnel in New York City at rush hour. It's, you only got 2 lanes to go through, and all this traffic is converging, trying to get into New Jersey, and it just can't go because it's all bottlenecked up, and the cell literally has a rough time of things. So these results then indicated that it's not simply sufficient to supply or attach an amino terminal signal sequence to a polypeptide to ensure its export. And, that was the great promise of biotechnology in the late 70s and early 80s, is we would use bacteria as factories to manufacture all of these proteins. Cloning was new in the late 70s, and it offered this great opportunity to make all these highly valued proteins. And so then Oliver and Beckwith came along, and they then describe, and the title of the Sec-A discovery was an E. coli mutant pleiotrophically defective in the export of secreted proteins. And pleiotrophy is a fancy SAT word, and it simply means when one gene influences two or more seemingly unrelated phenotypic traits. And so pleiotrophically, the E. coli defect was actually in the secretion machine and not in any of the proteins that were destined to go out. And so using Basford's original construct, the maltose binding protein LAC-Z, they reasoned that if they were able to isolate a mutant defective in components of the translocation apparatus, the machine that sent things out, that would leave a fully folded maltose binding protein LAC-Z fusion in the cytoplasm, they would then see it as enhanced beta-galactosidase activity, which would allow them to screen for positive lactose metabolism. And the way they did that is Beckwith was a pioneer of using these colorimetric assays, and the plate that he always loved to use was lactetrazolium plates. And so lactetrazolium plates, if you ferment lactose on a lactetrazolium plate the way Beckwith would make them, is you would be a very deep red color on a background of white lawns. And so if your protein was stuck in the cytoplasm, it would be very deep color. If your protein was stuck in the membrane, that is, you were gumming up the machine, it would be less red. And if it literally wasn't getting out at all, you then had no beta-gal activity. And so it was this clever way of asking the question, can you ferment lactose? And you pass your waste electrons rather than to lactic acid, you pass them to tetrazolium, which when oxidized is colorless and when is reduced is red. And how reduced it is how many electrons you give it. It gets redder and redder with time. Hence, you're able to spot on your lawn your organism of choice. So they could literally screen millions of colonies on a Petri plate and literally find that one lone mutant. And it was just an elegant way of pulling something out.
Nothing like a good screen.
And, it really defined bacterial protein export. Beckwith's group and Don Oliver was one that pioneered a lot of this work. Tom Silhavy did a lot of work on the outer membrane of gram-negatives. To this day, Tom still does a lot of work on the outer membrane of gram-negatives. And Basford was a pioneer until his untimely death. Phil Basford was on the faculty at the University of North Carolina. I remember going to a meeting as a freshly minted assistant professor and meeting him and talking with him in 1990, and then he was tragically dead in 91. So reading this, I sort of had tremendous deja vu just reading this very short snippet and reliving all of those fantastic, studies that were done in years past. And, as I said earlier when Vincent and I were talking, I said, reading these classic spotlights are like eating peanuts. You start and you can't stop. So I went back, and these have been published since January. And this particular one is in the August, or is it the August or July? Can't remember. Let me take a peek. It's in the August edition of the Journal of Bacteriology.
Right.
And so I went back and looked at some of the other titles, and they're just incredibly rich of what they have out there. Victor Rita, who's a friend of Vincent's and mine, he published one of these classic spotlights on phage bring punch to the party. Mm-hmm. And it's literally about diphtheria and how they discovered that the diphtheria toxin is on a phage. And it has the classic express train delivery of diphtheria antitoxin. And so that's a really neat one to go. And that's in the January issues. But the second snippet that I'm going to introduce you to is the other one that was in August. This one's entitled Studies on the Low Calcium Response of Yersinia pestis Reveal the Secrets of Plague Pathogenesis. And so this is a paper published 58 years ago in 1958, also in July. So I guess I'm picking the July papers. And since we are in the summer blockbuster movie season of zombies or apocalyptic movies, I thought a good old-fashioned paper on plague, a zoonotic disease transmitted by rodents via flea bites that caused 3 very large pandemics and were responsible for the dark ages on our planet. So the principal question that this classic spotlight paper offered us, and this one was authored by Olaf Schenwein from the Department of Microbiology at the University of Chicago in Chicago, Illinois. So the principal question that he presents to us, what is the basis for the deadly consequences of plague infection? Well, Lee Kuffenberg and Kiyoshi Higuchi studied the nutritional requirements of Yersinia pestis back in 1958. It was then called Pasteurella pestis, after, of course, Pasteur, before they renamed it for Yersin, who worked a lot on plague. And the title of the 1958 paper, which was not a paper, but simply a note in the Journal of Bacteriology. And so they literally are bringing out this one-page manuscript that was significant. And literally, because of this one-page manuscript that was published in 1958, I'm going to steal my own punchline, but from this one-page manuscript in 1958 came out type 3 secretion. And it's really a fascinating story to just sit back and see what actually then were the foundational studies that ultimately gave us our understanding of type 3 secretion and how it influences virulence. So 1958, Kuffenberg and Higuchi, their paper was entitled The Role of Calcium Ions in the Stimulation of Growth of Virulent Strains of Pasteurella pestis. And it's been reported earlier that year in a full paper in the Journal of Bacteriology that virulent and avirulent strains of Yersinia pestis exhibited markedly different growth rates when cultivated at 37 degrees in aerated, liquid-defined media. They further investigated this, and they revealed that the presence of a very small amount of calcium, 4 millimolar, in the medium was important for the growth of virulent strains of pestis at 37 degrees. And this requirement for calcium was not observed at 27 degrees. And they did this by classic viable counts, measuring things and seeing how many grew per unit time. And since 37 degrees is recognized as the temperature at which growth occurs in vivo or infections, its correlation between virulence and the requirements for calcium were may be very significant, or so the authors stated in their presentation of the data. And when they tested it in a mouse model, they found that doses of 1,000 bacteria or less resulted in the death of an animal, while the avirulent strains failed to kill at this range. And the authors then noted that the virulent strains of pestis consistently lost their virulence upon serial transfer in liquid medium, but they didn't see the same loss of virulence on solid surfaces. And they postulated that solid surfaces, because there's more calcium in solid surfaces, it would effectively hold on to whatever was the positive selective pressure to maintain virulence. And so what Olaf Schmidt presents in his review is this notion of we didn't yet know about plasmids. This was pre-plasmids of conferring virulence. So this is 1958 now. We barely knew about DNA. So it's really interesting watching how this goes about. And so the medium lacking calcium was found to be associated with virulence in a mouse model of plague disease. And we know that pestid variants that do not display this low calcium response, or LCR phenotype, are avirulent and were readily isolated when virulent strains were propagated at 37 degrees without calcium. So to me, sitting in 2016, that sounds like it's a plasmid in which you remove the selected pressure, and so the microbe doesn't like carrying that extra genetic baggage of the plasmid, and it kicks it out.
Yeah.
And so what then happened, now we jump forward to the 80s, and following this discovery that Yersinia pestis calcium dependence virulence was dependent upon a 70 kilobyte plasmid, transposon mutagenesis was used to then identify the genetic terminus of this low calcium response. And then, of course, Olaf then volunteers 2 other classic JBAC papers, and these are the papers that revealed that these plasmid carried genes that were the result of the transposon-based mutagenesis promoted the secretion of Yersinia outer membrane proteins, or YOPs, by a pathway that we now know is type 3 secretion. And that's principally the work of Thomas Michaels and Guy Cornelis, where they first demonstrated that YOP proteins accumulate in large abundance in media of Yersinia cultures propagated without calcium, and that this secretion again occurs via signal peptide independent mechanism. Because today we know that type 3 secretion is independent of the sex system. You can put out signal peptide independent simply because it's got a specialized secretion system.
So the calcium was just a lead to finding the type 3 secretion apparatus, right?
Absolutely.
Yeah. That's the key to virulence, the type 3 apparatus.
Right.
So cool. I like these spotlights because they're pretty short, and you can get a nice handle on the entire field starting from that one classic paper, They're quick.
And the editor actually puts it all into perspective to you. So I was thinking about, pulling the paper, but the graduate students will figure this out quick. Because if you pull the original paper and say, okay, what do you, to do the experiment of see how well graduate students are read, if you give them the original paper and you say, where can this paper take us today? And then you give them this classic spotlight paper and see, this is what happened from that one paper.
But if you went to that original paper, say as a student who doesn't have a lot of background in the field, it would be really hard to know where it went, without a lot of literature searching. But this puts it all there for you. It's really nice. Yeah.
And, I think, Tom Silhavi was really spot on with the issue of how people read today. I know in my own field, simply because there are so many journals out there, it's just so hard to scan table of contents of all the journals, all the stuff I'm interested in. You and I do it routinely because we're looking for cool stuff to talk about on TWiM.
Yep.
But I think the majority of our colleagues are just reading in their own area just to keep current.
No, I just, that thing about podcasting is makes you read outside your field. I really like that. So these are basically snippets. So I think Jay Bacht was listening to TWiM and they decided to make a written snippet based on
They did. They did.
Now, is this going to only happen this year?
I don't know. I think what we have to all do is we have to send Tom Silhavi a note telling him, yes, we love this feature.
Yeah, I think it would be great to go on for a long time. Of course, you have to get people to write them. And if you're not familiar with the field, it's not easy to do. So these individuals know the field. You could have written the SEC paper, the SEC snippet yourself, right? Having known it. But not everybody can do that.
No, and I don't know whether or not Vic Dorita, I have to send him a note asking him, did he know all that stuff about Corynebacterium diphtheriae before he wrote his little, we'll start calling them what we call them, snippets, as opposed to classic spotlights.
Yeah, I like snippet better. In fact, I can imagine these being the basis of snippets on TWiM. They're perfect.
They are indeed perfect.
And I think there's nothing wrong with going back 50 years and looking at a paper and what it led to. We don't have to always have papers that are in the last few months, right?
No, and the lessons learned, and you could ask yourself, this is how they did the technique then. How would we do it today? Because, some of these classic brute force cloning methods that Beckwith's team had to do were just elegant, and they simplified so much work. Today, we would just, clone it up. And, I was walking down the hall today, and there's a bunch of these ads on the bulletin boards for cloning services. Oh, yeah. You can just have stuff made.
All kinds. It's good to take away that time sink that, making clones in libraries. It doesn't give you the data. Doing the screens is what's important. So I don't mind that so much. But today you do all kinds of different things. You do systems analysis where you generate tons of data, and you do big screens, and it's not often for a while that you know what it means, unfortunately.
No.
Very different.
So
that's very cool.
As we transition to your paper, we're going to move into this brave new world of the microbiome for this next paper. And you wonder what people are going to be saying 58 years from today.
Yeah, it's, Alio has been very critical of many of the microbiome papers.
Yeah.
We do for good reason. I mean, we don't know exactly what they mean. And we're going to have to see in 10 years where these end up. But looking at them today, they're very appealing because if they're right, the consequences are really big.
Especially for the one that you're talking about today.
Yeah, this is really a remarkable example. I mean, in all the microbiome papers we've done, there are enormous implications of these bacteria that we live with. It's really incredible. They seem to control almost every aspect of our lives. And proving that is hard, but, we'll see. Hopefully we'll be around for 50 years.
58 more years.
No, I probably am not going to be, but... You
never know. You never know. Medicine is, you'll be double your age.
Yeah, I would be over 100. I don't think I'm going to make 100, but some people do. I'd be happy to hit the 80s,
Yeah.
That's okay.
Our role model is Dr. Schechter.
Dr. Schechter's going strong. Mm-hmm. Yeah. Okay, so here's our paper. It's a cell paper, and the title is Microbial Reconstitution Reverses Maternal Diet-Induced Social and Synaptic Deficits in Offspring. And the authors are Shelley Buffington, Gonzalo Viana di Priscio, Thomas Achtung, Nadim Ajami, Joseph Petrosino, and Mauro Costamattioli. These individuals are from the Baylor College of Medicine in Houston, Texas. And this is actually a paper that Michelle had pointed out before ASM Microbe. She said this might be a cool paper to do at some point, and it is really, really interesting. So this paper is about autism spectrum disorder, which everybody's heard about. ASD. This is a range of conditions that are classified as neurodevelopmental disorders, and they include features like social deficits, communication difficulties, stereotyped repetitive behaviors, sensory issues, and even cognitive delays. And, everyone knows that ASD is on the increase. It just goes up and up and up, and we're not sure what's going on. So there's a lot of research into what's causing this. Now, there is some epidemiological evidence that maternal obesity during fetal development increases the risk of a number of neurodevelopmental disorders, including those that fall within the ASD categorization. It's about a 1.5-fold increase if you are obese during pregnancy, 1.5-fold increase in the risk of these disorders. So in this paper, this is what the authors are looking at. They use a mouse model, and they ask, if we make pregnant mice overweight, what does it have to do with ASD? Now, I'll tell you in a moment how you study autism in mice. Obviously, it's not, you can't do the whole spectrum, but you select some things that you're able to study. So what they do is they take female mice and they put them into two groups. One group, they get a regular diet, and the other group gets a high-fat diet for 8 weeks. And what this does, it makes the mice fat. They clearly show that. They're now obese, and then they mate the mice, both groups, with males, and they have babies, of course, and the babies are given a regular diet. The babies are the offspring, so now we have offspring from normal mothers and obese mothers. They all have normal weights, so having an obese mother doesn't automatically make you obese, at least in this model. All right, then they say, let's look at social interactions. That's in autism spectrum disorder. That's one of the things that's deficient. There are many things, as I pointed out earlier, but that's one thing. And you can measure social interactions in mice. And it's very straightforward to do. And you can measure sociability. And let me tell you about this, because this is not something we talk about on TWiM, sociability, although we are very sociable, right, Michael?
We are.
If you meet us at ASM Microbe. So I'm going to tell you about these assays. Let me get them up here in the materials and methods, which are fortunately placed right with the article. Okay, so one of them is called the reciprocal social interaction. They put mice in a plexiglass arena. It's 25 by 25 by 25 centimeters, and they've never been in this before. It's brand new, and they either are put there with a familiar mouse, a cage mate, or a stranger, a mouse that they've never seen before.
That happens to us on airplanes all the time.
That's right. Okay. And then they look at them and they say, how many times do the mice touch? Do they follow each other? Do they touch? Nose to nose sniffing, nose to anus sniffing, or crawling over or under each other. Mice do all these things, right? You've seen mice in cages. They love to crawl over. And in fact, mice that have been together for a long time, they're always interacting. Okay. And so the person who's observing this writes down the number of times that these things get done. And they don't know which mice are in the cage. They're blinded. Okay, so they don't know that whether it's an offspring of an obese mother or not. So that's the social interaction test. Then they have a 3-chamber social test. Okay, this is called the Crowley's 3-chamber test for sociability and preference for social novelty. Okay, so you put, again, the mouse in a plexiglass arena. It's bigger. It's 60 by 40 by 23 centimeters. And this arena has 3 equally sized interconnected chambers. There's a left, there's a center, and a right chamber. All right. So then you can measure sociability, during which time the subject can either interact with an empty cup or a cup containing a stranger. And again, you ask how much time do they spend interacting with either the cup or the stranger mouse. And so this, you put the mouse in, and if it goes to a chamber with a foreign mouse and interacts with it, that's good, and so forth. So you can measure the effect of being born to an obese mother on these activities, their social interactions. Okay. So what they find is that the offspring of obese mice had impaired sociability, and they showed no preference for social novelty. In other words, if you give a mouse a choice of someone they know or someone new, they don't have a preference. But the normal mice born to normal mothers, I should say, like to explore new mice, so they're very sociable, and they like novelty. So in other words, you're born to an obese mother, you have social deficits. Of course, that's one of the characteristics of autism spectrum disorder. So that's why this is a nice model for autism and its origin, at least part of it, because it's reproduced in these social deficits. Okay, so that's the bottom line to begin with here. Now let's explore this a bit. It's been shown that maternal obesity can alter the gut microbiome, not only of the mother, but of offspring. And furthermore, kids with autism spectrum disorder have dysbiosis of their gut microbiome. It's not normal, as you might expect. So what they did is they looked at these offspring. They looked at the mothers and the offspring. They looked at their fecal microbiome by 16S ribosomal RNA sequencing. And of course, Elio would be quick to point out that this is not the gut microbiome, right? It's the fecal microbiome.
That's true.
Nevertheless, that's what they do because, well, if you want to look at the gut microbiome, it's harder, and maybe in mice you have to sacrifice them. I don't know. But anyway, that's what they did. So the obese mothers, as they expected, had a change in the microbial composition and diversity of the fecal microbiome. The offspring had very typical microbiota dominated by Bacteroides and Firmicutes, but there were differences in the structure of the community in the offspring from normal versus obese mothers, and the diversity of the fecal microbiome was reduced in the offspring from obese mothers. So just being born to an obese mother changes your microbiome. And presumably that's partially because of the influence of the mother on the microbiome that the babies acquire. Now, they then do an experiment where they take these offspring, again, from normal or obese mothers, they co-house them. You put them together, and, mice eat each other's feces. They're coprophagic, so it's like giving them a fecal transplant in a way. And this
much easier to do than us.
That's right. This corrected the abnormal social behavior of the mice born to obese mothers. So co-housing corrects the abnormal social behavior, and it also returned the fecal microbiome of these offspring of obese mothers to normal. This is the way this paper goes. It's really remarkable that at every step, it's exactly what you would like the results
infectious.
to be.
It's infectious.
It's amazing.
It literally is following Koch's postulates.
That's right. Okay, so what they're thinking now is that the lack of one or more bacterial species must be responsible for this abnormal social behavior that we can measure with these interesting cage experiments, okay? What they find is, looking over their 16S ribosomal sequencing, is that, all right, I'm jumping ahead. First of all, if the lack of a bacterial species is responsible for abnormal social behavior, you would predict that germ-free mice are defective in their social behavior as well, right? And
that's what you would think.
You would buy, so you can buy them. They did, and in fact, they put them through their social interaction tests, and they're also deficient socially. So that's good. They can then feed fecal microbiota from normal mice to these germ-free mice, and it restores their social behavior. Now, so that makes perfect sense, right? Because the normal mice have normal behavior, they have a normal microbiome. The interesting thing is, it works for giving them fecal microbiota from 4-week mice, but not 8-week-old mice. So if you feed these germ-free mice 4-week-old feces, corrects their social behavior. If you feed them feces from 8-week-old mice, it doesn't work. So there's some kind of window, what they call a neurodevelopmental window, for improving social behavior. This has to happen early in the life of a mouse. Otherwise, it may be too late.
And people, too, because we know autism spectrum disorder in people, there's this window in which it happens.
That's
and
right.
then it seems there's no return.
That's absolutely right. Okay, so what bacterial species are missing in these offspring of obese mothers? That can then go back and look at their data. It turns out the most reduced species in offspring of obese mothers is Lactobacillus reuteri, L. reuteri. Okay, this is a very interesting finding because it's already known from other work that this particular species promotes oxytocin levels. Oxytocin is a hormone that has a number of roles. One of them is it plays a crucial role in social behavior. So they take some L. reuteri and they feed it to the offspring of obese mice. So these are obese mothers. They give birth to mice which have social abnormalities. They feed those offspring L. reuteri for 4 weeks. This improves their sociability and their preference for social novelty. There's no effect of feeding L. reuteri on normal offspring. And if you kill the L. reuteri, it doesn't work.
Because you're not making the appropriate material.
Yeah, I presume these bacteria are making some kind of metabolite, which is, the key here. And that's going to be, I guess, a future paper.
The lactobacilli make lots and lots of things.
Yeah. And, Michael, they tried other lactobacilli, and they don't work in terms of correcting these social behaviors. For example, repetitive behaviors, which are part of autism spectrum disorder, are not reversed by the microbiome. And one of the ways they assess that is by a test called marble burying. Okay, it is the marble burying. They place mice in a cage with 20 marbles. They're black marbles, and they're sitting on wood chips. And then if the mice have repetitive behavior, they try to bury these things. And they go crazy burying them. So then they take the mice out, and they measure how deep the marbles are. And, so this is part of ASD. And that behavior is not reversed. So that behavior is caused by being born to an obese mother, but it's not reversed by giving them L. reuteri. So it's complicated, as you'll see, that autism has a number of different components, and maybe each one has to do with a different part of the microbiome. All right, so what's going on here? We know that L. reuteri is important. L. reuteri has some kind of connection with oxytocin. All right, oxytocin modulates aspects of social behavior, and interestingly, previous studies have shown that its levels are increased by L. reuteri. Oxytocin is made in the hypothalamus. That's part of the brain, a very small part of the brain that links the nervous system to the endocrine system via the pituitary gland. And in particular, the paraventricular nuclei of the hypothalamus make oxytocin. And from there, it goes to the pituitary and gets released into the blood where it has its effect. They looked at offspring of obese mothers. They looked in this part of the hypothalamus by immunofluorescence. They have fewer oxytocin immunoreactive neurons in the hypothalamus compared to the offspring from normal mothers. They're making less oxytocin. If you give these mice L. reuteri, it restores the production of oxytocin. So again, the bacteria must be making something that is somehow allowing proper synthesis of oxytocin, I would think, because oxytocin levels are going up. All right, now, oxytocin, the neurons that make oxytocin in the paraventricular nuclei, they go to another part of the brain, which is called the ventral tegmental area, and that's I believe that's part of the pituitary gland or near it. And that's one of the brain regions that responds to rewarding stimuli, and it's involved in social behaviors. All right, so oxytocin activates neurons in this ventral tegmental area. Let's call it VTA. So the oxytocin is made in the hypothalamus, and then it goes to the VTA, where it then influences behavior. And there have been a lot of other studies previously showing this. And in fact, in particular, if you blockade the receptors for oxytocin in the VTA, this in rodents prevents social attachment. So then they want to know if these mice that are born to obese mothers, do they have signal transmission issues in the brain? As you know, nerve transmission occurs across synapses. There are neurotransmitters that move, like dopamine, that move across the synapse, and they induce a current on the far side of the synapse. So they can use electrophysiology to ask whether social interaction causes these synapses to fire in the VTA neurons. Remember, the VTA neurons are the target of oxytocin. So basically, they allow mice to interact with each other, and then they sacrifice them, and they make slices from the brain, and they put electrodes in and measure the currents across into the VTA. So in their control mice, that is mice born to normal mothers, social interaction with a stranger, but not a familiar mouse, triggered long-term potentiation in these VTA neurons. Potentiation simply means the triggering of a nerve potential. In offspring of obese mothers, social interaction with a stranger did not induce this current in VTA neurons. So this is called long-term potentiation because you allow the mice to interact, and then you sacrifice them, and then you prepare a slice of the brain. You can still see the effect of that in the brain slices. Okay, so the idea here is that the oxytocin in the correct setting is somehow causing neurotransmission to occur, and you can measure that in the target of the oxytocin, which is the VTA neurons. All right, so the conclusion is that social interaction induces a long-lasting increase in the activity of the reward system of normal but not obese offspring. So you can see that in terms of behavior as well as in the electrophysiology of what's going on. Okay, how about L. reuteri? We know so far that Lactobacillus reuteri restores sociability and preference for social novelty in these offspring of obese mice. Does it rescue the changes in synaptic transmission in the VTA? And the answer is yes. They found that live but not heat-killed Lactobacillus reuteri rescues stranger interaction-induced long-term nerve potentials in the VTA as well as reciprocal social interaction. So not only does L. reuteri restore social interactions, but it also restores the neurotransmission to the VTA that is potentiated by oxytocin. So you got both social behavior and electrophysiological behaviors. So that's a little tough. I know that, but...
So the next experiment that we take from the snippets of earlier is we do transposon mutagenesis of L. reuteri and then feed the different strains and ask the question, which transposon mutant elicits the behavior or fails to elicit the behavior? And we can then at least initially pinpoint it in the chromosome of those L. reuteri and figure out what the gene is.
So what size experiment would that be, Michael? How many mutants would you have to look at? Thousands?
It depends on how you start with the shotgun. Remember, some of the early secretion systems were based on minutes of the chromosome, and there's 100 minutes of the chromosome, and they were doing like 2 5-minute chunks. They were doing this on the order of transduction mutants, which are about 100,000 base pairs. So it really depends on the genome complexity of L. reuteri. But you could we now know a lot about what neuropeptides are, and so we could do the genome scan and begin to target it. But,
could you take, maybe you could compare initially reuteri with another lactobacillus, maybe the metabolome, right, and see...
And ask who's missing.
What's missing or what major differences, and maybe use that as a beginning point.
There's enough sequence out there that you can do the old-time transposon mutagenesis without using transposons.
Yeah, right.
But this is so fascinating. I mean, even though they're starting with stool and it's not the intestinal microbiome, I think the brain slices would convince most people that there is something indeed going on.
Well, also you can feed the mice L. reuteri and it corrects the defects, right?
Yeah.
And then the last experiment they do, which everyone should be thinking about because we're talking about oxytocin as the messenger of all this. What happens if you give mice oxytocin? Well, they did that. They give it to them intranasally, and it restores the social interaction and the long-term potentiation in the VTA neurons of offspring of obese mice. Okay, so it improves the social interaction and it gives you the neurotransmission that you expect. So the conclusion is that offspring of obese mice have impaired oxytocin-mediated synaptic adaptations in the VTA that underlie social behaviors. So that ties it that's a sentence from their paper. That ties it all together. You need oxytocin to cause this neurotransmission, and in the absence of L. reuteri, that doesn't happen, and it's accompanied by aberrant social behavior. So right now, actually, oxytocin is used to treat autism spectrum disorder, but it's transient. It only lasts a couple of hours, and you'd like to have something more durable. So obviously, the idea is now that maybe you can give individuals certain bacteria. So L. reuteri would be one of them. They do point out that others have shown that other aspects of ASD in mice, other behavioral abnormalities, not the social ones, are corrected by different lactobacilli. So
incredible.
Maybe we're going to have a cocktail at some point of bacteria. I mean, this is at some point within the next 5-10 years, probably going to go into phase 1, I would guess. You just have to.
The problem is with human subject boards. They're very concerned about pregnancy and the long-term consequences for the obvious case. So, how do you do those sorts of experiments ethically is really a great discussion for an interprofessional ethics class.
Well, it's a tough one because the question is, do you treat mothers or children, right? And when do you treat them? At what age? Because there is a window in these mice, right? So
there's probably a window in humans, I think, guess, from everything that we've been learning about autism spectrum disorder.
So there's a lot of evidence that this makes sense in people. As I said, obese mothers are 1.5 times more likely to have a child with autism. The risk of children with ASD is two times greater when you're both obese and you have gestational diabetes. So this mouse model, it is a mouse model, but it makes sense in terms of what we know in humans. And finally, the last issue I just want to talk about briefly. Remember, L. reuteri, they think somehow promotes oxytocin-mediated functions. They think the vagus nerve is probably the path of communication between the gut and the brain. Remember,
the gut-brain axis.
Exactly. The L. reuteri is in the gut, but it's having effects in the brain. So how does that happen? Well, there's a nerve that goes from the gut to the brain. It's the vagus. The vagal nerve fibers project to the paraventricular nucleus. Those are the oxytocin-producing cells. So they're probably making a metabolite in the gut, which is traveling along the vagus and somehow is stimulating oxytocin production. Oh, that's incredible. Isn't that amazing?
It gives a whole new meaning to this bacterial communications. It makes quorum sensing look like Morse code.
Exactly. And finally, last experiment that others have done, if you do a resection of the vagus nerve, if you cut it, then L. reuteri has no effect on oxytocin production. So you need the vagus. So that seems pretty good evidence to me.
And it's, again, fulfilling sort of Koch's postulates. You cut the root, you're eliminating the ability to send the signal to the acting organ.
Michael, we once didn't even have a clue how many bacteria were in us. And now, without them, we'd be toast.
Oh, God. You wonder how we even survived the era of antibiotics and how we didn't just all die off and become the zombie apocalypse. Yeah, it's absolutely fascinating. And you wonder if some of these neurological diseases that we're seeing are actually a consequence of our outstanding nutrition of the 21st century.
We're holobionts. We're all holobionts. It's just incredible. incredible organisms. All right, this episode is brought to you by Curiosity Stream. I want to tell you a little bit about what they have to offer. They're the world's first ad-free nonfiction streaming service. They have over 1,400 titles, 600 hours of content. This company was founded by John Hendricks from Discovery Communications, and you should know that. And if you've watched any Discovery programs, you know that it's all about real science, not reality TV shows that currently plague cable TV. You can watch Curiosity Stream programs on the web, on Roku, Android, iOS, Chromecast, Amazon Fire, Kindle, and Apple TV. It's available worldwide in 196 countries. What do they have? They have a wide variety of science and technology content. They also have nature, history, interviews, and many other topics. It's all nonfiction. That's why I thought this advertiser would be of interest to our listeners. In addition to documentaries, they also have interviews and lectures. They have Next World with Michio Kaku, the future of technology. I searched for viruses. They have a few videos on viruses, one about Zika virus and the current epidemic. Inside the Human Body, exploring multiple topics related to the body from creation to survival and the brain. Microbirth, a documentary that looks at research that links the way babies are born with health and later in life, leaps in evolution, and much, much more. I think you'll like it. Monthly and annual plans are available, starting at $2.99 a month, $2.99 a month, less than a cup of coffee, or the cost of one title on a competing platform. For curious minds who never stop asking why, check out one of the largest 4K libraries on the internet. Check out curiositystream.com/microbe. Use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 months free of one of the largest 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign-up. We thank Curiosity Stream for their support of TWiM. Let's do a couple of emails here. We got one from Anthony, who sent us an article on how Dr. Arthur Conan Doyle cracked the case of the tuberculosis remedy. This is an article at pbs.org, NewsHour. And this was published on May 21st. It's written by Howard Markle. Sunday, the Sunday that this was published, marks the birth of a man who created the world's greatest detective, Sherlock Holmes. And he's reprising one of his favorite detective mysteries. Within a day after analyzing the clinical data, Doyle came to a startling conclusion. The whole thing was experimental and premature. While the rest of the world rejoiced over the reported conquest of tuberculosis, Doyle argued that Koch's lymph might remove traces of the diseased tissue but left deadly germs deep in the invaded country. Its real value, Doyle asserted, was as an admirable aid to diagnosis, and that a single injection would help doctors decide definitively whether a patient was in any way tubercular. By the way, there's a Koch mystery that I've been working on so far without success. A Google search on Robert Koch noise pollution yields a number of quotes like these. "The day will come when man will have to fight merciless noise as the worst enemy of his health," Robert Koch. Or the Nobel Prize winner Robert Koch predicted in 1910 that one day man will have to fight noise as fiercely as cholera and pest. The quotes seem to have appeared first in the popular press mid-century and now in journals. Did Robert Koch actually compose these statements? I don't know.
I don't know either.
But there's an article here in European Heart Journal, and it says right at the beginning, the Nobel Prize winner Robert Koch predicted in 1910, one day man will have to fight noise. So, I don't know, Anthony. I'm not familiar with this. Perhaps some of our listeners may know, but it sure is interesting. Our next letter is from Wink, a physician in Atlanta. He writes, "Michael, if the woman with the MDR UTI had not traveled recently, perhaps her intimate contacts imported the resistance gene to her. As a former Navy physician, I think this is a worthwhile epidemiologic consideration." Now, this was the first report of this MDR gene in the U.S., correct, Michael? That you
the colistin resistance gene that was plasmid mediated, yes.
So he's suggesting she may have gotten it sexually transmittable, right?
Or she could have eaten it, because remember, we learned that you can actually bring in E. coli from your food, and she may have ingested it because our food supply is indeed global, and depending upon what she ate, it could have. So it is indeed a worthwhile epidemiological consideration. It's not only could be an STD, as honeymoon cystitis will as reports, but it could also be ingested because the digestive tract communicates quite well with the urinary tract.
Yep. Our next is from William, who writes, "Greetings, TWiMers. I am working my way through the TWiM episodes, going backward in time, and recently listened to Elio's fascinating discussion on organelles. Thoroughly enjoyed his lecture on the nitrogen cycle as well. I just ran across this, and so pass it on just in case you haven't seen it. Quote," this is an article from Science Daily, "The first eukaryote is thought to have arisen when simpler archaea and bacteria joined forces. But in an opinion paper published June 16th in Trends in Cell Biology, researchers proposed that new genomic evidence derived from a deep-sea vent on the ocean floor suggests that the molecular machinery essential to eukaryotic life was probably borrowed little by little over time from those simpler ancestors." So that's a paper on the archaeal origins of eukaryotes. Yeah, that's pretty interesting stuff.
Well, we're getting better and better at looking at genetic drift with sequencing comparisons, and I think we're just going to have to continue to do more of it to get at some of these fundamental questions.
Low-key archaeum. I have a feeling that we talked about this, didn't we? I think Elio might have.
Yeah.
That's this archaean found on near deep sea vents. Has more eukaryotic signature proteins than any other prokaryote. Importantly, among these are proteins critical for eukaryotes' ability to direct traffic amongst the intracellular compartments. It sounds good, but you never know.
Yeah.
Thank you, Bill. Kayla writes, "Hello, TWiM team. I am a veterinary microbiologist working full-time in a veterinary diagnostic lab in Cork, Ireland. We had 2 lovely weeks of sun, but we're back to the rain again. I did my undergrad in the University College Cork and only graduated this time last year. I have finally, after months of listening, caught up with TWiM. And can I just say, these podcasts are absolutely wonderful to listen to while I pass my hours in the lab alone. They have helped me keep up to date with all the things micro, as I would otherwise have completely isolated from the outside world, be completely isolated from the outside world. One of my favorite absolute episodes is actually episode 10, a symbiotic cloaking device, all about Vibrio fischeri and the bobtail squid. I've listened to this story so many times, and not just from TWiM, but my lecturer in college who got me really excited about marine microbiology. I'm hoping to, in the not-so-distant future, go and study in Bremen and do the Marmic Masters. Wish me luck. I have so many questions, but they are escaping me at the moment. I will email them in the future with questions. Thank you for the wonderful podcasts. Keep up the awesome work. Time to tackle the other Twix episodes. Kind regards, Michaela."
Well, that was an episode by Margaret McFall-Nye.
That's right.
And I remember that. That was one of the early ones. Episode
10. You bet. Yeah, that was a wonderful story. Margaret used to be with us.
Yeah.
And she then left and hasn't come back.
Well, she went on sabbatical, and I think the squid got her. She literally fell into the lab and never got out.
She went back to Hawaii, I heard. Isn't that true?
Yeah.
Next email is for you, Michael. Supti writes, "I have a question related to microbiology. What is the difference between antibiotic sensitivity and antibiotic susceptibility?"
Sensitivity means that they are sensitive or will be inhibited by the, he's playing the game with the words, susceptibility and sensitivity. It's an inside baseball type of answer, and I don't want to get it wrong because I'll get letters. So let me stay tuned and I'll give you that answer because I don't know if I have everything right in my head at this point in time.
Okay.
I didn't do my homework ahead of time.
It may not be different, right?
It may not be, but the antibiotic sensitivity and susceptibility. Let me do a quick look.
All right. Well, I would say there's no difference, but what do I know? We'll come back. And if anyone knows, in the meanwhile, send us a letter.
Send us a letter.
And the last letter for today is from Kyle, who writes, "Hi, Twimmers. Thanks for all the wonderful podcasts. They've gotten me through many long days counting caccolithophores down here in Chile. Your descriptions of potentially daunting topics are spot on, as evidenced by the breadth of people who write you emails. You are all making very complex scientific ideas relatable to the layman woman with a basic scientific background, and it's really fantastic. Just one quick potential topic. In my undergraduate days, I did some research at a small rainforest station in Costa Rica. While I was there, one of the principal arguments I heard in favor of preserving rainforests is that rainforest diversity could lead to new antimicrobials, medicines, etc. Is there any basis to this argument? I noticed a dearth of rainforest-related TWiM episodes, and given the diverse microbial ecological communities in rainforests, I think this would be a fascinating topic for a future TWiM. By the way, one of my favorite TWiM episodes so far was number 107, in which you talked about the isolated indigenous community and how their gut microbes were susceptible to many types of antibiotics. Do you think saving some of these microbes for use as probiotics following antibiotic treatment could confer susceptibility in the remaining microbiota? Or are antibiotic-susceptible strains already used in probiotics? Thanks for all the bacteria for thought. Kyle is from Occidental College in California, where he says it's winter here, so the high today is 11 degrees Celsius, 62% humidity, 11 kilometers per hour wind, and 30% chance of precipitation.
That's because he's in Chile.
He must be.
Remember,
that's right. He's in Chile.
He's studying cocci that eat rocks.
That's right.
Coccolithophores.
Got it. All right, rainforests.
The reason we need to keep the rainforest is chocolate. Without the rainforest, there would be no chocolate. It's a natural fermentation that involves bacteria and fungi, and we haven't been able to make an inoculum that can actually do the wonder that is chocolate, because they take the cocoa beans and they ferment them in the jungle in situ, and we haven't been able to figure out how to make chocolate. And it's a paper from about 10 years ago, I think, or maybe a little bit longer. So we may want to pick that. But I agree, we need to dig out some papers on the importance of the microbes in the rainforest. And there are indeed new antimicrobials that we can pull out of the dirt, and in fact, of the complex rainforest. And as we move up and down the canopy, there are different, if you will, microbiotas in the canopy. As you move up and down the canopy, you can get different organisms and different communities. So I think it is an untapped potential, but unfortunately, folks aren't. They think that they are able to make these screening libraries, and that you can make molecules in the lab, and the screening libraries are all they need. They don't realize that the microbes can actually make a lot of these drugs, and they're much smarter at making chemicals than the chemists are. So
I know a lot of companies used to have natural product screen programs where they'd go out and get dirt, and a lot of them can them.
Yeah.
But, so I'm sure people are looking in rainforests. It's just that these typically don't get published, especially not where we look for papers for TWiM.
No, and a lot of the stuff the companies keep very, very close to their vest, and they patent it, and they may not release it, and it probably will never get published.
That's right. Michael, do strains used in probiotics, are they already antibiotic susceptible?
Some are and some aren't. It really depends on the probiotic. You can buy some probiotics that have bifidobacter that is sensitive, and then you can have others that are resistant. And so it really depends on where the probiotic is, being targeted for. And
is it a problem to take probiotics with antibiotic resistance genes in them?
I don't think anyone's done that formal study. I mean, it's really about what you want the probiotic to do. And as we just learned in the paper that you can have many strains of lactobacilli, but unless it's the right one, it may not be doing the right thing.
Yeah. All right. Thanks, everyone, for your emails. we love getting them. You can send them to TWiM@microbe.tv. And this has been episode 131 of TWiM. You can find all the episodes at iTunes, at microbeworld.org/TWiM, and also at microbe.tv. You should consider becoming a patron of TWiM and all the other Microbe TV family of science shows. You can go to patreon.com/microbetv, where you could contribute as little as $1 a month. $1 a month, less than a cup of coffee. And you get all the shows of Microbe TV, TWiM, TWiV, TWIP, and many others. Or you can also go to microbe.tv/contribute, and we have some other options for you as well. Getting your contributions will help us to do other things with our podcasts. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, Vincent.
You must have nice weather there today, right?
It looks like it's going to storm. I just looked out the window. We're getting our 4 o'clock thunderstorm every day.
Really?
Yeah.
All right.
It's summertime.
It's summertime here, too. It's 28 degrees Celsius and sunny with some puffy clouds. No rain in sight.
We have the thunderheads coming up.
And how long does it rain? For a half hour?
Yeah, typically. Though, last evening it thundered for quite some time with some spectacular lightning. The ocean's pretty warm and it's boiling over, and it's been about 37 degrees centigrade during the day.
It's pretty hot. Wow.
It's pretty hot.
Wow. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and Chris Condayan and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. Find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.